Hello students. Today we will be taking questions from molecular basis of inheritance, and along with the questions we will also cover concepts from PCR, a very important rule in genetics, and we will take up questions from nucleosome. Okay. During thermal denaturation step in the PCR, which bonds break? Let's recall polymerase chain reaction. Right. Now let's see what is polymerase chain reaction. We'll quickly see if it will have, you will also have a recap from this. Okay. Now what is PCR? So PCR means you have a double stranded DNA. Okay. And it gives you product as, what are these? These are base pairing. I'm drawing base pairing. Okay. The red one is the newly formed DNA. Now this is what we have to make with polymerase chain reaction. So these are two different strands of DNA. Okay. Now in order to have base pairing take place, we have to separate them. Okay. Now in order to separate them, which bonds are these? These are hydrogen bonds. Hydrogen bond between different bases. For example, A with T and C with G. Right? So from here to here we will break by heat. This is called thermal denaturation. As you can see, hydrogen bonds are broken with thermal denaturation. Okay. Now what happens in the next step? Now in order to form a new strand here, okay, what is needed? We need a polymerase and we need nucleotide. Yes. Okay. Now let's make this out of it. Okay. Now this polymerase will sit only when something is here, so that polymerase can extend this. Okay. Now this is called, this short length of nucleotide is called a primer. Okay. Now in order for this primer to bind, these bonds have to be formed again. So that means there has to be a cooling step. See, heating was breaking these hydrogen bonds. So for forming these hydrogen bonds again, we have to cool it. Now this primer will bind here. Primer is nothing but a stretch of nucleotide which is complementary to this stretch of DNA. Right? Now nucleotides will bind, polymerase will attach these nucleotides and a new strand will form. Okay. And this is called polymerization, or in PCR terms it is called extension. So as you can see, in PCR there are three steps. One is thermal denaturation, where the hydrogen bonds were broken between the bases so that both strands separate. Then there is cooling step, which is called annealing, because there was annealing of the primer with the template strand. And then polymerization happens, which is called extension, because polymerase extends this by putting these nucleotide in place and having these bonds form. These bonds are again which bonds? Hydrogen bonds. And these nucleotides are attached by which bonds? One nucleotide with another nucleotide. This is a covalent bond. Okay. So this is whole PCR. This is PCR. You are now good at PCR. Okay. You should be able to solve PCR questions. So obviously the best answer is B. Alright. So let's move on to the next one. Now let me know in the comments if you have any doubts. We can take up this again. Which of the following is true? In prokaryotes, mRNA, rRNA and tRNA are transcribed by single polymerase Yes, this is true. P and A sites are present on the large subunit of ribosome. Okay, so there are three sites, E, P and A. This is aminoacyl, this is peptidyl, and this is exit. Okay, and these are present on large subunit. Yes, this is correct. In eukaryotes, sorry, I should not have created a tick here. In eukaryotes, RNA pol 2 transcribes hnRNA. What is hnRNA? It is heterogeneous nuclear RNA, which is precursor of mRNA. mRNA is messenger RNA. Messenger RNA means that it carries message for the whichever protein has to be synthesized. Okay. Precursor means that hnRNA is converted later into mRNA. Okay. So that means it's precursor. Okay. So this is also correct. Okay. In eukaryotes, RNA polymerase 1 transcribes RNA, yes, whereas RNA pol 3 transcribes tRNA, this and snRNA. Okay. So this is also correct. Now if you want to remember, so RNA pol 1, 2, 3, R, M, T. Okay, remember this. You will never get confused. Okay, 1, 2, 3, RMT. 1, 2, 3, RMT. So RNA pol, rRNA, 2, mRNA, mRNA or hnRNA, yes, and RNA pol 3, tRNA. Okay, remember this. This is how you will remember easily. So we can see that all four statements are correct. So the best answer is D. Alright, now let's move on to the next one. Which histone protein is not part of the nucleosome core? Okay, now we have seen in the cell division and we have also seen in the biomolecules. Okay, and in fact we have also seen in the cell structure and functions. Okay, chromosomes are made up of DNA plus protein. Now DNA is made up of what? Now we have seen this all before. DNA is a polymer of nucleotide. Nucleotide means what? A nitrogenous base and sugar and phosphate group. Okay. Now phosphate group has what charge? So in order for this nucleosome to be stable or chromosome to be stable, protein has to have what? Positive charge. Okay. Now we see from the biomolecules in proteins which are the amino acids which are positively charged or basic residues. These are arginine and lysine. These are positively charged. Okay. All right. So nucleosome is a core made up of how many molecules? 8 molecules. 8 molecules total. Okay. And which are those? So H2A, H2B, H3 and H4, right? And all of them, what you are seeing in the front, are also at the back. So 4 here and 4 behind, so total 8. Okay. Now a DNA, okay, now let's say DNA. Now a DNA wraps around this Okay. Now obviously you have to imagine that this is wrapped around. Okay. Now I am drawing on a 2D surface, so that's why it's not clear. But you have to imagine that there is a ball, and when you wrap around a string around the ball, this is how the structure of a nucleosome will be. Okay. Now from here all the way this to here, how many base pairs are there? 146 to 147. In your NCERT books of biology, it is mentioned that 200 base pair, which is not true. Okay. So, or they are trying to say something else. And here is H1 present. Okay. So H2A, H2B, H3 and H4, these 4 molecules, okay, 4 types of protein and 8 molecules form the core, and this H1 is the not part of the core, okay, and it binds with the linker DNA. Okay. Why linker? Because this DNA again goes and form this, and there is another nucleosome there. Okay. Now this is another nucleosome. Okay. This is how it works. Alright. So let's see which histone protein is not part of the nucleosome. Not part of the nucleosome core. Okay. So these 4 are core. This is non-core. Best answer H1. Alright. Let me know in the comments if you have understood and if you have got this question right. Okay. Now let's move on to the next one. So nucleotide analysis of a 12,34,567 nucleotide long DNA sample has revealed to contain adenine 27%, guanine 23%, cytosine 29% and thymine 21%. It can be concluded that, okay, so see, whenever you see these percentages of these nucleotides or nitrogenous bases, always remember this is related to which rule? Chargaff's rule. Okay, that's your trick or tip. This is your tip. It's a very important tip. Chargaff's rule has to be utilized whenever they talk about percentages of different nucleotides. Okay. Now we know from base pairing that A binds with T and C binds with G. So that means now this obviously means that this DNA is a double stranded DNA. So in double stranded DNA, A concentration is always equal to, T concentration, and C concentration is equal to G concentration. These brackets are in concentration. If you want to learn more about concentration, I'll put a video here. You can check that out. Okay. We can see from the concentrations here that A concentration is not equal to T concentration, right? Because A is 27 and this is not equal to 21. So that means that this is not a double stranded DNA. How do we know it's a DNA? Obviously it's a thymine, so it's a DNA, right? So the best answer is, now let's see, it's a double stranded linear DNA. No, it's not double stranded. Doesn't matter linear or circular. Single stranded linear DNA. Okay, this looks like good answer. Double stranded circular DNA. No, cannot be double. Data is insufficient. No, we can deduce. So see, I have seen that this is my best answer, but still I have moved on to see whether other options are more relevant. Okay. So always, whenever you have questions like this, always look all the options and then you select the best answer. So in this case our best answer is B. Alright, it is a single stranded linear DNA. Alright, that's it for today. Stay safe, have fun, study well. Bye bye. Let's see you on Friday.